Aloha, my name is Katelynn Perrault and I'm an assistant professor at Chaminade University of Honolulu here in Hawaii. Today I'm going to be talking about decomposition odor research, how it's been evolving in the last little while, and the impact that COVID-19 has had on research efforts in this area. So I wanted to start by sharing my experience in a volunteer-led search that I participated in last year for an elderly missing person. It will come as probably no surprise to you that as a forensic scientist, we know that we recognize we're often dealing with the most challenging and sometimes traumatic events in people's lives. In May of 2020, we spent over a month looking for a missing person in one of the most expansive hiking trail systems here on the island of Oahu in Hawaii. The system is about 1,100 acres in total and has over 15 miles of intertwined trail systems with many additional minor trails interwoven in between those. I got involved in this search because the search lead reached out to me with several questions about odors that they were experiencing within the trail system. Scent detection canines were being brought in daily with no definitive results. Enough time had elapsed that the hope of finding a happy and healthy victim were becoming quickly diminished. And for the family, they were living through some of the worst moments of their lives daily while we traversed through countless tracks in the vicinity. And this satellite map shows the extent of searching and some of the key points of interest where evidence and odors were identified. It's in moments like this that I'm reminded that my field of forensic chemistry is actually rather unique and still very much so under development. The questions that I was being asked about the state of the body versus potential odors that could be produced could sometimes be answered based on our research, but not always. More often than not, I was giving answers like, well, it depends. And with deep gulches, steep terrain, thick brush, dangerous cliffs, and lots of decomposing wild pig carcasses, this search became strenuous and frustrating for many community members. Eventually, the victim was found resting in a plot of land that would not have been discovered without the key input of volunteer searchers, expert search teams, and other community members with unique knowledge such as myself. This took place at a time when the COVID-19 pandemic was impacting the entire world. May of 2020. For myself, seeing a community pull together against the additional challenges that this pandemic brought, beyond the typical challenges of a missing person scenario, made me hopeful. It certainly made me look inward to our discipline, how we're conducting research, the impact that the pandemic will have on those research efforts, and where we should be moving forward in the future. So today, that's what I want to talk about, where this field of research has been developing and where it's going to be moving, and how some of these impacts from the pandemic will affect things. So decomposition odor profiling is our topic, and the first question might just be, what is this field? Well, in this field, we look at volatile organic compounds, or VOCs. These are basically just chemicals that prefer to be a gas rather than a liquid or a solid, and they are being produced throughout the entire process of decomposition. So it changes over time from the point that the person dies to the point when they're discovered. And so the body is changing and this odor is changing with that. And we can think of odor actually as a forensic trace. It's probably not the first thing that comes to mind when we think of forensic traces, but it can be exploited and used as evidence in particular cases. So some of the applications of this trace would be to help understand and improve cadaver detection dog training, where dogs are used to search for and locate missing persons, victims of homicides and mass disasters. It can also be used to better understand how insects are attracted to a body throughout the course of decomposition, since this pattern can be used to help understand time since death. It can also be used to demonstrate the presence of a body, and there's actually been a couple cases in the US and in Europe where decomposition odor was collected as evidence in a case and used to show that a body was present even after it had been moved to a different location. There's also a lot of interest in using some of these target molecules that we detect in decomposition odor profiling to develop handheld detection devices that could be used in victim recovery. And finally, it does help us to better understand what's actually happening as these remains are decomposing. So how the soft tissues are decomposing eventually to the point where remains are skeletal. And while that's all very helpful, profiling this odor is actually very challenging from an analytical chemistry point of view. So I wanted to go through some of the challenges here. First, decomposition odor is a complex mixture of hundreds, if not thousands, of compounds all mixed together. These compounds also exist in a variety of different compound classes, and from an analytical perspective, it's difficult to develop a method that's able to detect all of those compound classes at the same time. There's also a broad dynamic range, meaning that some compounds are very high in abundance and some compounds are very, very trace in amounts. And having an analytical method that can detect both of those concentration ranges can be really tough. We also need to perform longitudinal analysis, meaning that we have to understand the odor as it evolves over time. So oftentimes we're dealing with many, many samples because we're collecting from the first point after death all the way to the end of the study. Also, this odor is impacted by many variables, some of which we'll discuss today. And finally, we do non-targeted analysis at this point because that's where this field of research currently is. We care about every single compound that's being produced by the body and trying to understand how it changes over time and with different variables impacting it. So since we want to know every compound that's there, that also makes our job as analytical chemists quite challenging because we need to develop a method that can target everything. So current studies have been performed in a variety of different ways, and this area of research has really been being developed since about 2004. The earlier studies looked at a single point in time, mostly from bodies that were both indoors and outdoors, but this looked at a specific time point. More recent studies have focused more on looking at this decomposition process and how it impacts odor over time, largely from bodies outdoors, because from a logistical standpoint, if you're going to be decomposing remains, having them outdoors has a lot of benefits. But in a variety of different scenarios, such as bodies which are buried, bodies which are deposited on the surface, say on soil or on grass, remains that have been submerged, and those that have more recently, in some studies, been placed under collapsed structures to simulate mass disaster scenarios. Studies have also looked at decomposition of both human and animal remains. The animal remains studies typically focus on Sus scrofa domesticus, which is a pig carcass. And some of these studies have been performed at taphonomic research facilities. Sometimes you hear those referred to as body farms, but these are outdoor facilities where both human and animal remains can be placed to observe and collect samples. So in collecting decomposition odor, the most common way of doing so is by placing a large stainless steel hood, which you can see in the photo here, and you place that over top of the remains. So this hood is sitting over top of a pig carcass. And what we do is we use a low flow sampling pump, which is essentially just a low flow vacuum. And we use that to withdraw air through a sorbent tube. This is a stainless steel tube that's on the inside. It has a sorbent, and that basically just acts like a trap. So as we pull air through that sorbent tube, through Teflon tubing, and then through the tube, our volatiles are going to be trapped on this tube, and then we can take that back to the laboratory for analysis. In the laboratory, there are two types of analytical instrumentation that are used to analyze decomposition odor. The first is gas chromatography, most often with mass spectrometry. And the second is comprehensive two-dimensional gas chromatography, or GCGC. One-dimensional GC is comprised of an inlet where we introduce our sample. The inlet is hot, so it makes our sample into a vapor. That vapor then gets swept onto a column, and that column is going to separate our chemicals from one another. Ideally, what then happens is our compounds will arrive at the end of the column where a mass spectrometer will be able to identify each of them and tell us how much of each compound is there. And that is how we build the odor profile. More recently, studies have started to use comprehensive two-dimensional gas chromatography, or GCGC. In this technique, we have all of the same components as one-dimensional GC. However, we have two modulator and a secondary column. What happens in this technique is as our compounds come off of the first column, or the primary column, they reach to the modulator, which is at point number 1. That modulator slowly traps and releases small plugs coming off of the primary column and sends each of those plugs onto a secondary column. It collects about 2 or 3 seconds of effluent from the primary column before sending that plug onto the secondary column, where it goes through a second independent mechanism of separation. What this means is that we are better able to separate our compounds from one another because every part of our sample goes through 2 different separations. And we still use a mass spectrometer to detect the compounds at the end of that process. So in GC, or one-dimensional GC, we have a plot that basically shows us peaks for each individual compound. So we can see a sample here. We've got 5 or 6 larger components and a lot of smaller components in this sample. With GC-GC, however, we are able to separate in 2 dimensions because we have 2 columns. And if we look at our plots, they tend to look a little bit more like a heat map, where each of these individual, we could call them blobs. That's actually the term that's used. But each of these individual blobs would represent an individual compound. And so we can see in this bottom plot here, there's still a big chunk along the bottom that's not resolved. But if we look to everything that's kind of above that area, we can see many, many components that weren't necessarily visible or separated in the plot above. So this is actually the same sample analyzed by both techniques. So compared to GCMS, GC-GCMS provides us with better peak capacity, and because we can pull our compounds apart from one another better, we have increased sensitivity and detectability of our compounds, and our compounds show up in these kind of patterns along the plot that allow us to be able to start identifying them more easily. This means that when we're doing decomposition odor analysis, we get more information about the sample. We have improved visibility over differences from one sample to the next. And therefore, we can actually just start to focus only on the compounds that are the most important in our samples because we can actually separate them from things like background. And that's important because there are so many variables that impact the production of decomposition odor. We have intrinsic variables, things coming from the body or the carcass itself, extrinsic variables like weather and climate that are impacting the body, analytical variables like the type of instrumentation that we're using, the type of maybe sorbent in our sorbent tube, so different analytical things related to the collection and analysis process, and taxonomic variables like how long has the body actually been there, and what scavengers are in the area, etc. And we have started to study a lot of these factors. And so on the last slide, you would have seen asterisks on different points where we've actually started to study a lot of these different variables. And from that, a lot of the studies have started to evolve. And I can say that the most effective studies have some things in common. First off, using animal models to develop different methods is really helpful because it allows us to have multiple carcass replicates. This is helpful because we can start to see how trends vary from one body to another. However, of course, it's always helpful and necessary for us to validate those trends that we see on human remains. It's also helpful when studies monitor decomposition odor over more than just a single point in time. Having a single point in time doesn't necessarily give us a full picture of what's actually happening. It's also helpful when representative control sites or background sites are used in replicate because this helps us to develop better statistics so that we can understand what's happening in our samples and differentiate the background that may be coming from things like soil or plants or building components that maybe are in the area that aren't necessarily contributing to decomposition odor, but nevertheless show up in the sample that's collected. It's also helpful to have analytical controls for collection and analysis. So for example, having blanks that help to show what volatiles are coming from the stainless steel hood, what volatiles are coming from the sorbent tubes themselves, etc. It's also helpful to use the appropriate analytical tool for the question at hand. So if a study is looking to perform untargeted analysis, GC-GC is going to be the better tool because we have a better ability to comprehensively separate all the analytes in the samples. However, if a study just wanted to look at maybe 5 or 10 volatiles and how they're changing over time, a one-dimensional GC analysis may be perfectly suitable. It's also helpful when studies report significant compounds in their samples based on statistical variance, i.e., how compounds change from one sample to the next, not necessarily abundance. If we look to a lot of the earlier studies in the field, researchers tended to focus largely on the compounds that were the most abundant, and we now know that some of the lower level compounds are actually very important in that profile as well. So over time, we've been developing more and more studies in this area of research. It started around 2004 where the first study was published, and now in 2021, there's approaching 50 published research papers specifically on decomposition odor analysis. I have a couple asterisks here, one at 2008 and one in 2011, and that's because in 2008, there was the first occurrence of a case where decomposition odor was collected as evidence in the case. In 2011, that case went to trial, and that decomposition odor was presented in court for the first time. This was at a time when there were fewer than 10 published studies in the field of decomposition odor. So I like to point out how far we've actually come from that point in exponentially increasing the amount of research being conducted in this field, and how much more helpful that information could be, to a case that's similar to that at this point in time. And if we look on the right-hand side of the slide, we can see how the techniques have been evolving. So the earlier on studies tended to focus mostly on one-dimensional GC, whereas now we start to see a lot more occurrence of comprehensive two-dimensional GC in these research papers. So what we know from a lot of these studies is that during the fresh stage, which occurs immediately after death, there are very few compounds present, and they tend to be lower in abundance. That's what we see here with low concentration of compounds and lower diversity in the fresh stage. Once the body starts to decompose, it becomes bloated, and we get a lot higher concentration of the compounds that are being produced, but still we don't have too many compounds present. The active decay stage, when there's a large amount of insect activity and a large amount of soft tissue loss, is when we have a peak in the concentration of decomposition odor, as well as the diversity in the amount of compounds we have. This is where the most compounds are typically detected in all studies. And then in the advanced decay stage, we start to see that those concentrations start to diminish because there's less soft tissue present. However, we do see the persistence of these at low levels, meaning that we maintain the diversity of those compounds that's detected in the profile. So again, just to cover the stages and what's happening, during the fresh stage, there are very few VOCs detected, all at low abundance. In the bloat stage, that number of VOCs will start to rise, but we have a lot of evolution of concentration of those because putrefaction is occurring. During active decay, the VOCs are at peak production because there's lots of putrefaction, liquefaction, and microbial processes happening. And in a lot of studies, we see thousands of VOCs detected at this stage, all at very high abundance. The advanced decay stage, we start to see the abundance of those VOCs reduce. However, the complexity of that profile becomes maintained. And during the dry or remains or skeleton stage, we reduce those number of compounds, and really the compounds that will be retained will be the ones that are in the environment that are the most persistent. So what we know about decomposition odor right now is that we have at least hundreds of compounds in about 10 different chemical classes, that this profile is dominated by sulfur compounds, namely dimethyl sulfide, dimethyl disulfide, dimethyl trisulfide. This odor starts minimal, it will peak in abundance and complexity, and then it will reduce. And it's impacted by many factors, including temperature and humidity and insect activity as probably being the three main environmental factors. The odor that we collect will be slightly biased by our collection methods. So if we use different methods of collecting this odor, then we may get slightly different profiles. However, there is a subset of this profile that is reproducible under different conditions in totally different environments at totally different times of the year. We also know that live odor transitions to post-mortem odor after about 43 hours or about 2 days post-mortem. What we still don't know in this field is the mechanisms behind how a specific compound is produced. So we can't necessarily track a starting point to an ending point in terms of which compounds are being released. And we also don't know how to predict the presence and the specific amount of a compound at a very specific point in time, meaning that we don't necessarily know if we could use certain compounds as markers of postmortem interval. We also still don't fully understand the impact of microbial communities on this odor. And we know that microbial communities have a very big impact on how a body will decompose. So that's still a big blind spot in this field of research. And because we still have some of these blind spots, we don't necessarily know how to interpret samples if they are collected in casework. So it would be best for us to develop a better understanding of this before, again, presenting this type of research in court. Looking to a map, these are some of the areas in which VOC studies on decomposition odor have been conducted. If we look at climate classifications, we can see that the large majority of these studies fall into the Köppen class C and D. The area that I'm currently located in is actually Köppen class A in Honolulu, Hawaii, where we're actually designated in the tropical rainforest category. So our most recent study focused on looking at decomposition odor of pig carcasses in this region and seeing how that can compare to other regions that have been studied around the world. This is our field site in Palolo Valley, which is in Honolulu, Hawaii. And we can see an aerial view on the right-hand side, a view kind of looking out towards what we call Diamond Head. If you've ever been to Hawaii, you probably recognize it. It's very close to Waikiki. And we also have a photo of some researchers outside at our field site taking some measurements from a pig carcass. So this is a type of terrain that we're typically looking at. It's very similar to a lot of the areas that people go missing, especially missing hikers, missing elderly people, etc. So it's very representative of the scenario that I started this presentation with. And in studying this, we monitored decomposition odor over a 2-week period, which is typically about enough time in Hawaii for our remains to go all the way to a skeleton state, or at least to advanced decay. In this, we were able to detect all major compound classes that we know exist in decomposition odor, except aromatics and alkanes. The acids, sulfur compounds, esters, and nitrogen compounds were the most significant components of the profile. The temporal trends, or the trends over time, appear to exist for most major classes in very similar ways to areas such as Greece, Canada, and Australia. And obviously, if we want to compare our study to other studies, we can only compare it to studies that have similar study designs, that monitor over a similar period of time, etc. And therefore, this list is really only this list of places that are similar is really only representative of studies that have done similar types of research or designed their study in a similar way. If we look at these VOCs by decomposition stage, we can kind of see them separated, and we see how the VOCs are lower in abundance in the beginning, and then they kind of peak and then eventually start to decrease. And the sulfur compounds did start to dominate that active decay stage, which is very similar to other studies. We see that the acids started to peak in later decomposition. Again, these trends have high consistency with similar data that are reported in Belgium and Australia. And so this is very encouraging to see that in a totally different climate, we can identify new trends, similar trends in new regions that are, consistent from one place to another. And of course, a lot of our research has been impacted by the COVID-19 pandemic. I wanted to give a bit of an idea of how this kind of impacted our work. For those who don't know, Chaminade University is a predominantly undergraduate institution. We're located in Honolulu on the island of Oahu, which is what people probably consider to be, the main island in Hawaii. And we've been, rather isolated by an ocean border throughout this pandemic. And so that's been a benefit in some ways, and in other ways, kind of a barrier, at least to our research. But in March 2020, we lost full access to the university. About 2 weeks into March was when that first happened. Very similar to, I imagine, what happened around the entire world about that time. For us, we were quite lucky that we actually regained access for research purposes only to the university in May of 2020. And this was because an exemption was put in place in our locale for research to continue. And so the university granted us access. However, we had to go through quite a bit of paperwork for every researcher coming back on campus. We were only allowed to have limited personnel in our space. Absolutely, it makes sense because of what was going on. We also had to have additional safety protocols in place that obviously is necessary, but also impacts your productivity. And we had to train everybody on these new protocols. And as well, we were also training new researchers in the laboratory on new techniques. And so that makes it really challenging when you're trying to train new people in the laboratory, but you have restrictions on the number of people you're allowed to have in that space. So it just changes a little bit the way that, you have to work. In August of 2020, we actually started our fall semester, and at Chaminade University, that was a fully online start to the semester. And so we did about 6 weeks of online studying, and then in October 2020, all of our classes resumed face-to-face. And we've been face-to-face ever since, with the option for students to study online if they want to. And I point this out because at predominantly undergraduate institutions, or PUIs, a lot of our research space is actually located in classrooms. And so that's a huge consideration, because through the summer months, we were basically able to maintain social distance and make sure that we had limited people in those spaces. But as soon as classes resumed, it became a lot more challenging to book time in the laboratory, to access the instrumentation, to access our data workstations, and things like that. So even though, classes and research at a lot of institutions are taken completely separately, for us, the start of classes, even online and face-to-face, actually did have quite an impact on our ability to get in there and do work. From a field standpoint, if you were to be conducting field research throughout this time, of course, impact to field facilities could be challenging. So first off, you have to make sure that your personnel get access to your field facilities. Typically, that involves preparing in a laboratory environment before going out to those field sites. You're talking about teams of researchers needing to access those facilities. So, you have to think about people driving together in a car to that location. By the institution, it may not be. There also may be an impact of availability of research specimens. So, for example, we get pig carcasses from a local pig farmer, and so that can be challenging if they're not willing to provide things throughout, these major incidents. In terms of funding, if you're working on an existing funding program, you really have to think about how are you going to make your funding program work given a totally new timeline. Granting agencies, of course, were very understanding of setbacks to research over the past year. However, if you're working on, a multi-year funding program, keeping researchers employed and keeping equipment running and maintenance going and everything, all of that is going to be impacted in the long term in how much you can really achieve throughout your funding program. And also, this area of research is an area where there's already very minimal funding, and there's been a huge uptick in the number of grant proposals that have been submitted across, I think, probably all areas of science right now, as people were, at home writing and compiling, previous data and things like that throughout the pandemic. And so that funding availability could also be an impact, moving forward, at least in the next year or two. In the laboratory, a couple of things that we have to think about as well. Obviously, I've already kind of talked a little bit about personnel access to laboratory, but I also wanted to mention for us, one of the things that was really challenging and completely understandable from my perspective was the level of commitment that my researchers were able to put towards their projects. My entire research group is undergraduate researchers. A lot of them have part-time jobs, they're also studying full-time, and they have family commitments outside of the university. And a lot of that really impacted the ability for us to have really high expectations of how much could be performed in a certain period of time. So we had to adjust a little bit the expectations about the commitment to, performing research hours, because these are part-time workers. We also had to think a little bit about, if we have team projects and multiple people working on things, how do we organize to have them in the laboratory at the right time? How do we make our workflow in terms of data processing and all of that work properly? We had to think about, how do we take data that's on our computers here at the university, and how do we make that accessible for researchers when they're working remotely and things like that. The equipment also had to keep running throughout this entire time. So we needed access to we actually had limited access to technicians. We had 2 incidents where we were supposed to have technicians come out to do, regular maintenance. And those things were definitely impeded as well. And being, for us at least, that we're an island in the middle of the Pacific Ocean, obviously we had challenges with shipping of supplies. And we also have to think about how we're using our funding, for example, because as we have to keep our instruments running with helium constantly, which is very expensive, if we're not actually running samples, we're still actually funding the use of that instrumentation even while it's sitting idle. So just some things that we had to kind of think about and plan for, at least as we move into an area of time where we hope things are going to start to return a little bit more back to normal. So in conclusion about this work, I would say that the field of decomposition odor is still relatively young. It's developing, but it's getting there, and we are learning so much more through every study about what this odor is, what the profiles look like, and how that can be applied in different scenarios. So we've learned a great deal about these trends and how stable they are in different areas. We still need some validation studies. Some is probably an understatement. I would say a lot more validation studies are needed to really put this into practice. Better modeling of this data that we've even already collected to look at trends, maybe looking at using some more advanced statistical models to try to pinpoint how this odor is impacted over time, and maybe even use it as a post-mortem interval marker. And also, I talked a lot about the different things that make a study the most effective, and I think implementing those across a lot of studies will be more helpful for studies in the future. And of course, COVID-19 will have an impact on most research groups to some extent, either in the laboratory or the field, or in both capacities. And so we have to also kind of understand what we're up against moving forward, and hopefully, moving more into a time that's more normal. And I just wanted to take a moment to acknowledge all of the researchers in my group. They are very hardworking. We've all been through a lot this past year, and it always amazes me every day that they continue to work so hard on their research projects. So I just want to take a moment to thank everybody. And also to thank the organizers of this conference. It's a pleasure to be able to be involved, all of our funding agencies, and of course, if you have any questions, you're more than welcome to reach out to me, and I've just attached my email to the bottom of this slide. Thank you very much.